What if we came up with a large scale experiment in our community on us streaming? Like, all right, we get like multiple cultivators, we test out the cultivation, co-culturing practices. I mean, imagine the freaking, imagine the authors on that research paper. That'd be a long list. That'd be fun. Yo, but I think we came up with I think that's a good idea of how we could, go about this research, y'all. As far as how to take even your own foraging practices right now, the next time you go out into the wild, taking those soil samples. How do you do that? I don't know if you guys are familiar with that. You can, just basically bring some sterile water with you and of like the wood, have it soak in there. And then when you can do serial dilutions and plate that kind of stuff and isolate the strains, the species that are around. I don't know if you guys are that technical, but you could be, and that'd be tight. And then maybe you could PCR it. You could, really identify what the species is. And yeah, we can get that like mapped. Like just how you see like common trees with certain species, like we'll be able to map the common bacteria and the common other fungal species in the area. That will give us evidence for what should be co-cultured. But then there's a whole nother variable side of it, because once we start adding bioinformatics to it, we can identify even a more technical level, machine learning, AI, on what is the actual connection between these, map that pathway even better, and start introducing maybe super far reach types of co-cultured strains or species. Like that's where we start taking marine species and introducing them to the terrestrial species. Like we could get some crazy stuff with that. You know what I mean? Crazy, crazy, crazy. So there's so much room. There's so many species that I don't even know. Like I don't know what Philinus, Orientaceae, like I don't study endophytic, mycorrhizal species really either, but I'm willing to now. And I think that we're not seeing that. By us not seeing that, it gives room for us, which we have other strains already. We have different strains. Maybe you guys are doing magic mushrooms. I don't know what you guys are into, or you're doing algae or something like that. Like co-culturing, y'all. That's the premise for all of this. All right. So induces multiple metabolites. Like, y'all, we're not just getting more potency. We're getting novel compounds as well. Crazy. All right, let me go back. Crazy. Y'all, so like I'm curious, like what are I think whenever there's like an unknown as far as like how to proceed with the research, where to start, you got to just look for nature to show you what they're already doing. You know what I mean? Because you always find species grown together. You always do. Like how many times you find a log and there's like a Ganoderma species with some turkey tail there too. Boom, we should be co-culturing those. How many of y'all probably have those two cultures right now in your strain inventory and can go do that right now? I bet you. Go make an LC and just plop both of them in there. I don't know. It's got to start raw at some point. You know what I mean? The research is going to be pretty raw. It's going to be pretty, it's going to feel whimsical at first. But when we apply the same scientific rigor and questioning and the same testing that would be done formally, then you're going to get some practical data from it. And we just got to keep going. We got to communicate just like we're doing right now. We got to get people talking in the chat. We got to build a community and get people doing this stuff. Co-culture of heterotrophic nitrification. I'm going to make a list right now, and I'm going to put it in the chat of the articles that we are finding, okay? That way you guys can go and read them on your own time if you have access to the paywall. And if not, maybe you can keep tapping in with me and I'll be talking about this kind of stuff. Okay, beneficial effects of this endophytic fungi. I gotta start getting familiar with some of these different fungi too, because I mean, it may seem practical for us to do like two big species that we see, but more often than not, you're probably going to be more successful in this doing a more macro and maybe like a super micro or endophytic type species. You know what I mean? You'd probably have more success in the sense that the complexity of those species, the dynamics that they're already in, may be outweighing the other. And so, although it may not have to be a competitive dynamic, it kind of is just based off of maybe the species, right? So maybe it might be helpful to have some type of endophytic fungi with a more macrophytic fungi. What about those like molds or whatever that, or alternative forms of certain species that exist within it? Like, isn't there like a mold that lives inside of cordyceps that kind of comes out when it gets too hot? I don't know if that's real. But just seeing something, if that is real, seeing a dynamic like that, that kind of gives evidence of like, yo, big and small. That may be like an immediate like starting point for co-culturing here. And that's why it's so important for us to do those more primary steps. It's going to be absolutely foundational for us to go into these practices by going into the wild and isolating the different species that are involved in that network, in that ecology, in that local ecology. You know what I mean? Because those are the pressures and dynamic relationships that are driving those expressions, those gene expressions, those proteins expressions. That whole environment. It's not just temperature, humidity, you know what I mean? It's multiple species. And what are we doing by this? We're alleviating metabolic stress off the organism, which allows for, perhaps that's what's needed for silent gene clusters to be activated, because maybe most of the metabolic energy is being directed to more triggered enzymes. You know what I mean? And some of the more complex, novel enzymes require more energy that isn't necessarily required to reproduce, which is typically the biggest driving force. So we have beneficial effects. Let's jump into this. Endophytic fungi possess favorable effects on their host plants, including disease resistance, improvement, secondary metabolite induction. Okay. Boom, right there. Right there. Right there. If you watch the Science News podcast, there was one of the papers was, I think it was episode 2, one of the papers was what is the compound that has been shown to be like the threaded like communicating signal between plants and certain fungi, right? Like when someone's like, they just yell out hello, maybe certain compounds are released by anyone, and that can be some type of receptor molecule. But then maybe I want to speak specifically to fungi as a plant, or vice versa. I'm going to send out a more specific compound, a chemical signal, to get that interaction going, right? To establish that connection, that bandwidth. I think it was like GLE16, right? I think that was the compound specifically. But in those cases, we see that certain you have like the more macro species, like a plant, and you maybe have the endophytic species, the more smaller one. There's signals going on, right? There's signals going on. There's compounds being directed. And that communication seems more common in a more non-competitive dynamic. If we're talking competitive versus non-competitive in this co-culturing for activating silent gene clusters to produce a variety of compounds, right? I noted earlier, like, all right, we want to get more antibacterial compounds from our fungi, co-culture with the bacteria. More fungal species or more antifungal agents, co-culture with the fungal species. From a competitive standpoint, that makes a lot of sense. From a non-competitive standpoint, maybe we want to aim for that macro micro dynamic, because those dynamics, macro and micro, it's like their reason for relying on each other seems pretty easy to understand or easy to establish. Like, yo, I'm the bigger species, I can protect you, hide inside of my roots, versus you're super small and right there next to those nutrients, you can start breaking them down and you know, fixing them into a more bioactive and bioavailable form. It's like the more primitive guy-girl dynamic, right? One's bigger and stronger, the other one is, you know, is more flexible with their tasks, right? It seems more easy or caring or dynamic like that. Like, it seems easy to draw the line. All right, you go do the physical as a man. Obviously, I'm not trying to say that is always the case. I'm just talking like primary dynamic roles. I know we're in 2025, things are different, but the point still stands, right? The point still stands that that dynamic seems very salient, and we should just use that. We should just use that as our entry point into this research, as a guide, right? Look to nature, right? Grab the low hanging fruit, see how that works. Let that validate this methodology, this philosophy we're establishing. So if we're looking for a non-competitive co-culturing dynamic, we're going to go with the macro micro co-culturing. If we're looking for maybe specific, maybe it happens even faster, we'll go with macro species. Or maybe you're doing micro species. And I know those aren't the correct words I should probably be using, but I mean it in the sense of like, all right, this one produces the fruiting body, this one doesn't produce a fruiting body. Macro micro, or like a plant endophytic fungi. You see how there's like one larger, obviously one, and one obviously smaller. Their abilities will be different, but that dynamic relationship, that local relationship, can still be implemented in vitro in the lab. So let's use that as a jumping off point. So you have species determination. And we said we'll go with macro, micro relationship. We got a macro. We've got micro. Honestly, we probably have more than just a dual relationship. Like, I don't want to start the next, the next plateau right here. Two species just seems very feasible and practical, but in reality, right, mycorrhizal species of fungi are probably, have relationships with bacteria and trees, plants, so to produce a fruiting body. So the relationship, who knows, maybe when we take these soil samples and we start to get a more clear picture of how diverse these local relationships are, whether competitive and non-competitive, both, that will start to be mapped to the various ceilings of concentrations and potencies and novel compounds that we're trying to reach, right? Maybe those will be associated in some logarithmic pattern based on the number of organisms in that local ecology, in that local relationship dynamic, that local, yeah. It's like, because they're shifting and maneuvering resources and protection and those things like that. So maybe, you know, axenic monoculture, we get this level of range of potency and level of compounds because the metabolic immune system and activity and stress is all dependent on one organism. We introduce 2, we reach a new teeter. 3, 4, 5. And they're not just randomly introducing random other species. These are species that have been validated based on the samples found when finding that local mushroom or that wild mushroom and isolating those strains and introducing them at different levels, different concentrations, different species, and seeing if there's some pattern as far as, all right, having these sets of organisms alleviates this range of metabolic of stress, metabolic requirements. This class of organisms does this role. And so when we introduce any type of variance in those classes of organisms, we'll still get that role fulfilled in alleviating the metabolic stress to produce more novel and higher potent compounds, biochemical compounds. So I don't want to create the next plateau by saying it's just macro micro relationship, macro macro micro macro, being like larger species, smaller species, exo endo, but it's practical to step into it just like we started with one culture, trying to just selectively have one. Now we can control, try to get to 2, and then more dynamic. And the layers that we add on will be indicative or will be backed by what we find from the isolated samples in that wild environment. We can be introducing more and more cultures in our co-culturing in order to alleviate additional stress. So again, when this research starts to evolve and we start to map some of these events and these pathways and these relationships, we'll be able to like almost like selectively doing it like a plasma map. We'll be able to be like, all right, now I can add in this species at this stage, at this concentration, and then it should activate this class of compound production, or you know what I mean? And then I add in maybe a marine type species or an extremophilic species, so I can have it withstand this acidic liquid broth or something like that. You know what I mean? We can get real deep and creative with how we start to not only mirror what's done in nature, but apply that variance, that intentful variance to that as well. You see what I'm saying? Yo, this is crazy. This is absolutely crazy. This is and it's not just random. It's not just random. We have rationale to direct this research. So we said species determination. And then what did I say just now? I was saying the variance. The variance. So yeah, that's. But we're trying to get to what? We're trying to get to the point where we can do what I just said, which was mix and match. Mix and match. Co-cultured species, mix and match culture species to activate various classes or various metabolic pathways that, so mix and match co-cultured species to activate various metabolic pathways that result in potentially specific classes of compounds. You see what I'm saying? And I mean that by like perhaps adding in. All right, so what do I mean by that? Like, I don't know, like a certain type of endophytic or bacterial introduction that maybe these bacteria are able to, they have some ability to rework some type of nutrient or tap into some type of nutrient and make it available in a local environment. Maybe it's a fungal species that's able to do it, right? Because they're excreting things. Maybe that's like another key to how we interpret, how we might discern these patterns, these abilities. So like, we want to mix and match the co-cultured species to activate various metabolic pathways that result in potentially specific classes result in potentially specific classes of compounds, result in the production and the potential production, result in the production. I'm such a stickler with words, y'all. So like writing, sometimes I get stuck in it, but it's important because it really holds on to the rationale, logic, thought process I'm trying to get out. So mix and match co-culture species to activate various metabolic pathways that result in the production of potentially hyper-specific classes of compounds. Perhaps classes of species will provide metabolic alleviation for our fungal species to produce more potent and novel metabolites. More potent and novel secondary metabolites. I guess I could show you what I'm writing, y'all. It's just Word. I'm just on Word, so it's nothing crazy. But and this is just me writing my thoughts out. Like, I'm not reading behind the paywall and doing that kind of stuff. This is just from our conversations, reading abstracts and stuff like that. So we're talking about co-culturing strategies. We have to understand what goes into those strategies. We're going to need species determination. The goal of co-culturing is to alleviate. The goal of co-culturing, or I guess the justification, that might not be the right word for co-culturing, is to alleviate the metabolic stress / load typically delegated. Oh, I can't even spell now. Delegated to our monocropped species. Now, our monoculture, our axenic culture, axenic species. Is that the right? Am I using axenic? I feel like I just learned this, so I'm trying to use it correctly. So axenic. Yep. The state of a culture in which only a single species, variety, or strain of organism is present. So it's an adjective. Yeah, so I'll use it as an adjective. Justification for co-culturing is to alleviate the metabolic stress / load typically delegated to our axenic species in vitro cultivation. Okay. There's other implications for this research, like, right, we're trying to find antimicrobial agents, right, antibacterial agents, about antibiotic resistance. So going into stuff like that is obviously additional means for it, but the justification for this logic, this rationale, to even look to co-culturing. Why look to co-culturing? It's to alleviate metabolic stress, which is typically delegated to our axenic species in vitro cultivation. Excuse me. Because, right, we usually make it sterile, quote unquote, and isolate just the one species that we want to grow to get, big yields and whatnot. But to get more compounds, we introduce co-culturing, and we vary the type and number type amount. We vary the type, amount, and number of species in co-culturing in order to selectively alleviate specific metabolic tasks or roles / processes. You know what I mean? Maybe these classes, maybe a specific classes of bacteria are always able to cleave a certain chemical that's involved, that's a rate limiting compound in the production of some beneficial compound, right? Maybe there's a process that creates this beneficial compound, and by increasing the other organism, by introducing an organism that can, it's like adding ribosomes. All right, remember we were talking about the catalytic ability of RNA, right? It's self-catalysis, right? But eventually that self-catalysis led to the amino acid or peptide synthesis, or this primitive creation of an enzyme, ribosome, right? That could then do that process better as far as like creating more things, right? So that may have just like not made sense, but hopefully it did. But we're trying to vary the type, amount, and number of species in co-culture in order to selectively alleviate specific metabolic tasks, roles, and processes, right? And who knows, by maybe introducing bioinformatics, transcriptomics, we'll be able to do this at an accelerated rate. You know what I mean? If someone's very skilled in that domain, that may be a more practical approach to get into this research. And that'll level up that side of this whole thing. Meanwhile, you got other people that have more lab equipment. They're more skilled on the like hands-on biotechnology, biotechniques. And that is their introduction and how they start this as well. So here, let me show you guys this. So right now you've been seeing this screen, and I am still adding to the list of articles. I'll add this one as well.